software download and interface. Now we will demonstrate how to use EasyCancer. Download EasyCancer for Windows, macOS or Linux from the LabSinal website at the Resource tab and then to the EasyCancer tab. On the EasyCancer page, a brief set of information about the software and detailed instructions of use is available. After downloading, open the file as an administrator. The EasyCancer interface should open. In the Initial Values panel, define the initial parameters for each cell, such as initial probabilities of division and death. Next, load the Easy table, which holds information about events, such as mutations, as well as the interaction table. In the Stop Condition panel, set the desired stop condition, such as the number of generations, the total number of cells, or the number of affected cells, which corresponds to any cell that contains one or more alleles affected in any gene. In the Tumor Environment panel, define a maximum tumor growth rate, which will eliminate mutated cells when the population growth rate is above the defined value, which can be derived from real tumor growth values. This function is optional. Finally, in the Simulation Parameters panel, set the number of cells in the initial population and the parameters to iterate the seeds, which allows the analysis of several tumor evolutionary stories in a high-throughput manner. Output files can be chosen for each seed, or for the entire simulation, or for both types. As all parameters are already set, next load the Easy table. An example is available on the LabSinal website. In the Easy table, the genome size is the sum of the oncogenome and the normal genome. The oncogenome is a list of genes that, if altered by a given event, with the defined probability, will have an impact on the Easy cell. For each event, a dominance value can be set, which corresponds to the impact of the event if it occurs in one allele. Alteration in the second allele leads to the full effect of this gene. Next, the probability of division and death can be set, which will impact the fitness of an Easy cell being hit by a given event, and thus affect their presence in the final population. Additionally, the number of events per division and the maximum number of divisions per cell can be chosen to, respectively, model the mutator phenotype and cell senescence. Finally, EasyCancer also allows to model the impact of tumor microenvironment, affecting the maximal tumor growth rate when this function is enabled. Besides, for each effect, the operation can be set as multiplication or addition. After the Easy table is loaded, two options become available. The Load Interaction Table button is an optional function, and one example can be downloaded from our website. On the interaction table, you can set the impact of a given event onto another event and the order in which this interaction is computed. Also, you can set the impact of the probability of division, probability of death, and maximum number of divisions per cell for this interaction. Through this interaction table, it is possible to model mutual exclusivity, in which an event has reduced impact on the fitness if a given event occurred previously, and co-occurrence, in which a given event has an increased impact due to the occurrence of a previous event. This table also allows the modeling of copy number variations, in which an event affects more than one gene. The second option available when loading the Easy table is the Manual Events panel, which allows the user to insert specific events at a given generation. This event can impact a defined percentage of the population, and if this event will happen only in one or both alleles. These roles can be edited and removed. Now that all parameters are set and the Easy table is loaded, simulation can start by pressing the Run Simulation button and selecting a directory for outputs.